Speaker is Dr. Nilesh, and he'll be speaking on biologics, biosimilars, and biobetters, understanding the basics.
Thank you so much, ma'am, and I would like to thank
This term is
AIOCMTC2.
becoming very fancy now. B, all
Yeah.
B.
So I would like to thank AIOCMTC Scientific Committee for providing me this opportunity. I'm Dr. Nilesh Kumar. These are my financial disclosures, and these are our contribution. We have been publishing for last 4 years. We have around 25 papers on biosimilars and biobetters. So this is the group led by my mentor, Dr. Ashish Sharma, and I was his resident for 2 years, and we had published around 20 papers at that time, and then we have continued the journey. So what are biosimilars? What is the public perception? The public perception is this. The original is Mohabbatein, and in a Kapil Sharma show, a skit is happening. That is biosimilar, a cheap copy, a generic medicine. But is it so? It is not like that. These are two different molecules. They share the same basic structure, they share the same efficacy, and they share the same safety, but they are two different molecules. So it is not a generic copy, as we see in a normal pharmaceutical, but it is a complex protein. By definition, it is highly similar to approved biologics, notwithstanding minor differences in clinically inactive component, having similar safety, purity, and potency. So every change, the primary amino acid sequence is same, but then there are post-translation modification, and that will lead to biosimilars. So how the biosimilars are manufactured? After the patent expires, actually it starts much before the patent expire, but for legal purposes, it starts after the patent expire. Reverse engineering is done of the proprietary molecule. That basic amino acid sequence is interpreted and kept the same. So if it is having the same, primary structure, but the secondary, tertiary, and quaternary structure might be different. The site of action should be similar to the innovator. That is a must. And cell line for production might be different. So what cell line was being used 5 years earlier, 10 years earlier, 20 years earlier, that might not be same, but it has to have the same purification. The preclinical and analytical studies to prove similarities with innovators. So a lot of these comparisons happen in lab rather than on patient. They do a spectroscopy and see if there is a similarity is established, and then large number of pharmacokinetic and pharmacodynamic studies are done to compare the molecule. So you can see for the innovator molecule, these studies are very limited. A small number of analytical study is done, or preclinical study is done, and most of the clinical studies are done on the innovator molecule. But for it becomes opposite for the biosimilar, where patients trial is very less. So across the globe, there is a big concern across the globe that only 3 months data is taken before the drug is approved as a biosimilar, and that might might make it cheaper and might make it unsafe. But the clinical trial design is for equivalence, and equivalence can be proven in 12 weeks, not more than 12 weeks. As we go ahead, the equivalence might differ. So in the first 12 weeks only, we have to prove the equivalence, but the safety trial and the monitoring, efficacy monitoring and safety monitoring goes on for 1 year. So the trial runs for 1 year, but equivalence is taken at 12-week endpoint. And this equivalence is taken differently in US FDA and the European. In US FDA, it is taken as visual acuity. In Europe, it is taken as central subfoveal thickness. So a receptor for all these diseases, this is also a extrapolation of indications. So if a biosimilar is approved for one indication, it can be extrapolated and used for all the indications that is being provided for the innovator molecule. Legally, it has to take, like Intas has taken for Razumab, it has taken for myopic neovascularization and all, but it is not mandatory, it can be extrapolated as well. What are the pros of biosimilar? Why do we need them? It is because the apparent reason is price reduction. There's direct discounting from the innovator. As we can see, we must have noticed all these innovator companies who were not budging their prices now are coming with various schemes. दो पे एक free, Eylea लो तो second one is free and then third के बाद fourth and fifth. Nothing was there till the biosimilar came. So that is the direct price benefit, and the indirect benefit is from the lesser disease burden. So socio-economically, the patient is growing, the disease burden is less. The inapparent is wider availability and the local pharmaceutical having better approach. So Novartis, like I am practicing in a tier 2 city, Novartis came to me for 2 times in last year, Allergan came 1 time, but the local Lupin and Intas is visiting me every month. So they are giving me greater grassroots penetration also. So is it all rosy? It is not. It can be very scary. As we can see, the innovation, as the more number of generics in antibiotics started, the innovation in antibiotics started to come down. We don't have any new antibiotic in pipeline because all the company now started focusing on generic and producing the copy of the same antibiotic, and now we are in a crunch where there is antibiotic resistance, but we don't have anything new to offer to our patient. So this might also happen in biosimilar, but there is a way out. What is the future? Like Dhoni is a biobetter of Kapil Dev. Kapil Dev ke time pe there was only one ODI World Cup. Dhoni won Champions Trophy also, T20 also, and he won ODI also. Similarly, there is no standard definition of biobetter, but biobetter is something that was created as a biosimilar, but is having either a longer duration of action, more potent action, or more purity, more shelf life. Any one aspect is better. That what we call as a biobetter. So who can benefit for biobetter? 2 molecules that were very good, very efficacious, abicipar pegol, couldn't receive the FDA approval, and brolucizumab, that is Pagenex, is not being used because it is having inflammation. So if we add a pegtail in the brolucizumab, it might lead to lower inflammation, and it will help us with, it can be relaunched again. So thank you for the opportunity. Thank you. Sorry for overs.
Till the time patent of Pagenex is there, you can't think of adding anything to your structure to minimize inflammation.
So,
What I'm saying is, you said in the definition of biobetter, abicipar as well as Pagenex. Yes, so can you today modify that?
Biobetter is treated as a new drug, sir. If they apply for a application, it will be considered a new drug. New drug.
You can't modify Pagenex now because they have that patent for at least.
Yes, sir. The company itself, they can modify it, sir. Not nobody else right now.
Correct,
Yes, sir.
Correct.